So again, welcome everybody. My name is Lakesh Sharma. I'm a soil fertility specialist and a BMP coordinator in Soil and Water Sciences Department, IFS. I'm the organizer of this BMP Summit along with Dr. Michael Dukes and Emily Eubanks. Thank you, both of you, for your help and support. We have two speakers today, Maria Silveira and Yiannis Ampatzidis. Sorry, my pronunciation is not very well. Forgive me for that. Maria is a professor in soil fertility and water quality. She'll go first, and Yiannis will follow up around 1:45. So Maria, the floor is yours.
Thank you so much. Thank you for the opportunity to present today. I'm going to share my screen. If you can please give me a thumbs up if you can see my screen. Okay, sounds good. Yeah, thank you for the opportunity. We only have 35 minutes for this presentation, so I'm not going to be able to provide a whole lot of details about some of the studies, but I'll provide some of the reference. A lot of the information that I'll be presenting today, it's been published, and also I'll share my contact information, so feel free to reach out to me if you have any additional questions. Here's a brief outline of my presentation. Before I even talk about the biosolids studies, I would like to go over some background information, why we call biosolids, what is the difference between sewage, sludge, and biosolids, how it's managed in Florida, and what are the regulations that govern land application of biosolids. And then very briefly, I'm going to go over two studies. One is completed, one is an ongoing study, and I'll provide you more information as we proceed in the presentation. So let's start with some definition. You probably heard different terms to refer to biosolids, and especially if you look at the public press, you may even see some terms with a very negative connotation. But the term biosolids was created in the 90s to differentiate materials that are untreated from those that are meet federal and state regulations to be land applied. So the biosolids are this can be a solid or semi-solid or even liquid material that's produced during the domestic wastewater treatment. So the sludge that is go to landfill, that is not a biosolid. That's a material that hasn't been treated to the extent that to meet state and federal requirements. So in Florida, how this material is disposed? So it's domestic wastewater, and there's no, in the US, we don't mix industrial and domestic, so we're strictly speaking about domestic wastewater treatment residue. So there's different disposal options, and today the reason why land application is highlighted is because most of my presentation is going to be focused on this land application as a fertilizer source or as a soil amendment. But as I indicated, biosolids can also go to landfill. Some material, if they go further, they are further processed, they can be distributed and marketed as fertilizer. If you go to Home Depot or any hardware store, you're probably gonna see class AA biosolids that are marketed and as fertilizing. A lot of homeowners probably don't realize that they are actually using biosolids, but yeah, it's a big proportion of biosolids in Florida are sold as class AA. They can be transferred to a bigger facility, usually smaller municipalities. They transfer their biosolids to a larger municipality, or can also be incinerated. So in terms of the proportion in Florida, currently we produce about 340,000 metric tons of dry biosolids per year. About 2/3 of that 340,000 is beneficially reused. And by beneficially reused, I mean they're either land applied or they're marketed as fertilizer. So the remaining 1/3 goes to landfill. Some of you might be familiar that the rules governing land application of biosolids was recently changed. So that proportion, this 2/3 of beneficial reuse, I suspect it might change So I think we're going to see a larger proportion of biosolids going to landfill in the future. So what are the types of biosolids? There are two types of biosolids, Class B or Class A. Sometimes, depending on the treatment, can also, in Florida, we call AA. Class B is the minimum, refers to material that meets a minimum quality for beneficial reuse. They undergo different treatments to reduce pathogens mainly, but because it's, like I said, is a minimum quality material, there's some site restrictions. So most of the Class B biosolids apply to pastures or citrus in Florida, and that's the majority of the land application in the state refers to this Class B material. So for instance, I work primarily with forages. There's grazing restrictions. When you use Class B materials, you have to defer grazing for a certain period of time to avoid direct exposure of grazing animals to the biosolids. Class A or AA are the materials that are marketed as fertilizer. Like I said, you can find Class AA in a hardware store, and those are meet a higher degree of treatment. So they essentially eliminate pathogens and is the highest quality for beneficial reuse. So the regulations in Florida are based on the type of material. If you're talking about Class A or Class AA or Class B. And also based on the composition, the nutrients and other pollutants that are present in the material. So in terms of regulation in Florida, we follow federal, state, and a lot of counties have their own local ordinances. So the federal regulation is often referred as Part 503. It was developed in the 70s, and it establishes limits for pollutants and some management practice associated with land application and disposal of biosolids. We also have state regulations, and I indicated early in my presentation that they recently went to a revision. So our current regulations look slightly different than they were in the past few years. And those state regulations were initially based on Part 503 on the federal, but they also included some additional requirements, mainly to control nutrient movement from each application area. So they are more restrictive than the federal regulation. And as I said, about 1/3 of the counties in Florida have developed their own local ordinances. So they are usually more restrictive than the state level regulations. So as you can see, there's multi-level of regulations that control land application of biosolids. So it's not something that either a municipality or agriculture operation can do without any guidelines. So there's a very strict protocol. Each facility that is receiving the material, it needs to be permitted. So they need to go through a very detailed permitting process. So I'm not going to spend too much time, but there's a number of benefits associated with reusing biosolids. They are a source of nutrients, macro and micronutrients, especially in our extremely sand soils. Because biosolids contain significant amounts of organic matter, they can provide multiple benefits to soil physical, chemical, and biological properties. Oftentimes, biosolids are used in reclamation of disturbed lands. So it's a very common practice in many mining operations to use biosolids as a reclamation strategy. And biosolids also conveying a lot of environmental benefits, one of them being carbon sequestration, greenhouse gas mitigation. When you compare the same amounts of, for instance, nitrogen added via inorganic fertilizer or biosolids, there's a potential for greenhouse mitigation. But of course, as anything, there's also some potential risks. And most of my presentation today is going to be related to this nutrient accumulation and potential subsequent impact on water quality. This has been a huge issue in Florida and in other states where biosolids being land applied. There's also concerns regarding pathogens, odors, and a number of contaminants of emerging concerns have been also raised concerns. So in terms of, we have to differentiate what are perception from what is really scientific evidence of risks associated with biosolids. And as you go, and I just want to mention this very quickly here, as you go through the literature, there's a number of citations and review papers indicating that the current federal regulations are adequate to protect human and the environment. So as you can see in this National Academy review that was published in 1996, it's a very detailed review that states that if biosolids are applied in accordance with the guidelines, they pose no risk to the human health or the environment. Subsequently, in 2002, there was another document pretty much reiterating what the 1996 review document indicated. There's no scientific documented outbreak of illness in humans associated with microorganisms in biosolids or any contaminant of emerging concern. More recently, I want to highlight this, and I'm not sure, don't worry if you don't have this link, I'll be happy to share this in the chat box later on. This is a report that was published in 2020 by a multi-state group that's called W4170. The name of this research committee is Beneficial Use of Residuals to Improve Soil Health and Protect Public and Ecosystem Health. It's a very, this group has a long-term history of biosolids research. This group, they were part of the Part 503. They provide the scientific basis for Part 503, which is the federal regulations governing land application of biosolids. And they, like I said, last year, they published this report in response to EPA report that was published by the Office of Inspector General. And the title of this report was EPA Unable to Access the Impact of Hundreds of Unregulated Pollutants in Land Applied Biosolids on Human Health and the Environment. So when the EPA, the Office of Inspector General, published this report, there was a lot of concerns that were raised regarding these unregulated pollutants. And what this group, this W4170 members of this group, what they did, they wrote a report in response to that EPA OIG report. And I would, if you are interested, I will encourage you to take a look at this report. And basically, they are using science-based information to address some of the concerns that were raised in this OIG report. So essentially showing that as long as the federal guidelines are in place and their biosolids follow those guidelines, there's no human or environmental risk associated with land application of biosolids. So I mentioned that the nutrient issues are particularly important in Florida because it can very easily disrupt water quality in our state. So in Florida, this rule 62-60-40 refers to the state regulations that control land application of biosolids. And the state regulations require two important things. One is the nutrient management plan, which I'm not describing here. It's a very detailed process. You have to have a number of information about the soil type, the materials, the sources of nutrients that are being applied to the area, what kind of crop you are growing. So it's a very detailed, you have whoever is applying for a permit have to indicate that information. In addition to this nutrient management plan, we also follow the Florida P index. I don't want to get into the details what the Florida P index is. Maybe other speakers in this summit touch into this Florida P index, but very briefly, it's just a site-specific tool to determine the potential risk of phosphorus transport from an agricultural field. So it takes into consideration the source of the P source that you're using, but also some site-specific characteristics. And so we use this P index to determine whether biosolids can be applied on a nitrogen rate or a P-based rate. What that means is if the P index, for instance, is low or medium, then the biosolids can be applied at a nitrogen-based rate. What that means is, let's say for a forage that requires 160 pounds of nitrogen to the acre, biosolids will be applied at that rate to meet the crop nitrogen requirements. However, if the site, and like I said, this is site specific, so it's for each application zone. If the P index value is high or very high, then the biosolids need to be applied at phosphorus base rate. So the crop P requirements, not the nitrogen requirements. And that's where things get a little bit of tricky, because when you apply biosolids at a nitrogen base rate, oftentimes if you repeat that for many years, you can provide excess P, which may not necessarily be an issue, but depending on the site, it can become an environmental concern. On the other hand, if biosolids are applied at P base rate, oftentimes that's impractical, because that means that the producer will have to supplement nitrogen with a different source, probably inorganic fertilizer. So that becomes very not economically feasible in many situations. Another issue related to nutrients and biosolids is the potential for over-liming. That was an issue, especially a few decades ago in the 90s and the early 2000s, where the majority of the biosolids were treated with calcium carbonate to eliminate pathogens. So the lime stabilized materials had a very high pH, and producers were using high rates to supply the crops with nitrogen, and they forgot about the issue with the pH. So once the pH of the soil raised above a certain level, many crops cannot perform well under that high pH condition. That issue has been addressed in our state regulations, because now that they are required to measure calcium carbonate equivalent. So just a way to compare what would be the liming potential of that material. One of the issues that we deal with oftentimes, the environmental hazard associated with phosphorus in biosolids has been perceived as being greater than any other P source. In this case, I'm mentioning here fertilizer, manure. And that philosophy, unfortunately, goes against most of the scientific literature. And I'm going to show just a few examples here, but there's a very extensive literature showing that, in fact, the phosphorus biosolids P availability is much lower than any other P source. So when you look at this data published in the early 2000s, it compares phosphorus solubility. This is the y-axis, a percentage of water extractable P. So the greater the water extractable P, the greater the water solubility. And it compares against different P sources. So TSP is triple sulfur superphosphate, so commercial inorganic fertilizer, versus dairy manure, poultry manure, and two types of biosolids. As you can see, most of the phosphorus present in organic fertilizer is soluble. So you apply, if the plants are not able to utilize it, you would accumulate in the soil has any phosphorus holding capacity, or it can be subject to losses. By comparison, you can see the phosphorus solubility in biosolids are significantly less than any other source, any other P source. However, if we're trying to differentiate, biosolids they can go through different treatment process. And depending on the treatment process, that phosphorus solubility can also be affected. So this next slide shows, this is data from Dr. O'Connor, where he compared different types of biosolids and the percentage of water extractable P. In the previous regulation, the biosolids type was not differentiated. Now we have a new tool that allows for greater application on materials that have very low phosphorus solubility. So what I'm trying to say is, biosolids phosphorus solubility is less than any other P source. However, depending on the treatment process, you may end up with a wide range of materials. They're all called biosolids, but they can behave differently in the soil. So to address that concern, going back to the outline, I'm gonna very quickly mention about this first study that we did using a rainfall simulation to examine the difference in phosphorus availability among different biosolid sources. And like I said, this is the reference. This paper has been published. It was published last year. And so the goal was to, as I said, just characterize this phosphorus solubility across different biosolids materials. What motivated us to do this study, and we were focused especially in the St. John's River water basin, was because this particular part of the state has experienced a tremendous increase in biosolids application for various reasons. There's some other parts of the state where biosolids application were banned. So a lot of biosolids that were produced in South Florida end up being land applied in the St. Johns River basin. And there was some indication that there were some water quality issues that the same area was experiencing. The question was, is it related to biosolids application, or it could be something else? So that's the main motivation why we decide to conduct this study. So we selected materials that were the main biosolids material that were applied to this region, and we decided to run this rainfall simulation study. And these were the predominant biosolids material that were applied in this area. What I want you to just quickly glance is the treatment process. So most of the Miami-Dade produced biosolids material were anaerobically digested. So we call this the Broward, the Miami, the Tampa, and Green Edge and Bradenton and Sun Beach. We call all this what would be the traditional biosolids. So it goes through either an anaerobic or aerobic digestion, and you end up with a material that has very low phosphorus solubility. The only material here that's slightly different, and it has nothing to do with the class, as you will see in the result. Class of biosolid has nothing to do with the potential environmental risk associated with phosphorus transport. The only different material here was this biosolid that was produced in Orlando, and it's called a biological P removal process. So that's the BPR stands for, this acronym. Usually this biological phosphorus removal process results in biosolids with a high phosphorus solubility. So we were concerned whether this material could be causing some issues, and that's why we include that. And a significant proportion of biosolids that were applying that St. John River basin was this BPR material. This is the analysis of the material. I know it's a very crowded slide, and I'm not expecting you to take to look at the data, but what I want to show is that depending on the treatment process, you can also have different characteristics, different nitrogen to phosphorus ratio. So the study covered that wide range of materials that were applied in this region. And the reason why we use this rainfall simulation is to mimic what will be the worst case scenario. So we pack soils in these boxes, and we decide to, because we're mimicking what will be a land application in a pasture situation. So we establish a grass cover. This is Bahia grass, which is the most common forage crop where biosolids are land applied. And then we surface apply the material, the different biosolids material to these boxes. And then we simulate a specific rainfall to generate runoff and leachate. And this is the rainfall simulator in operation. So we have just what we call double deck boxes. We can collect surface runoff. We can also collect leachate. And very briefly, this is the result. So this is flowweight runoff soluble reactive P, is the mass that was lost via runoff as affected by the different P sources. What I want to highlight is this first bar here. The blue bar is the control. So it's just soil with no P application. And this large yellow bar here, I don't know if you can see my mouse, but the yellow bar refers to the inorganic fertilizer. So we run this rainfall simulations 3 times, and this is the flow weight concentrations, P concentrations in the runoff. As you can see, inorganic fertilizer resulted in a much greater runoff P than any other P source. With the exception of this light blue material, that's the Orlando or the BPR material, all the other biosolids result in similar same runoff P concentration as the control. So basically it was not statistically significant than treatments that received no P application. Again, with the exception of this Orlando material. And when we look at the leachate, pretty much the same story. So this is leachate soluble reactive P as affected by P source. No differences among the different biosolid sources with the exception of the BPR material, and inorganic fertilizer resulted in much greater leachate P losses. When you look at the percentage of phosphorus that was added that ended up being lost, of course, as you can see this red circle here, about 40% of the phosphorus that we apply via inorganic fertilizer was lost in runoff or leachate. But again, this number cannot be translated into a field situation because there's no opportunity for the plant to utilize. So we apply the P sources and we made this huge, we created this artificial rainfall to collect runoff and leachate. And there was a strong correlation between, so how can we predict which materials will result in greater leachate and runoff P? And phosphorus extractable P in the material has been, so this percentage of water extractable P has been a common indicator of how soluble that phosphorus is in the biosolids, and our data just confirmed that statement. And in fact, this is now part of the requirement. So anyone interested in land application of biosolids need to determine this percentage of water extractable P in the biosolids material. So there's a strong, the reason for that is because there's a strong correlation between that number and what can be potentially lost in leachate or runoff. I'm not gonna go into details, I'm just restating what I said. Biosolids represent a much lower risk for phosphorus and nitrogen losses as compared to inorganic fertilizer, but the results from the study cannot be translated into field conditions because it should be interpreted as a worst case scenario. We had wet soils, there's no plant uptake, and we created this intensive rainfall immediately after application, immediately after the P source application. So we decide to do something, well, if it can provide some information, but cannot be applied, information cannot be directly applied to sites that are receiving biosolids. So we decided subsequently to conduct a field study. And this study has been in place since 2016. The first 4 years was funded through the Florida Cattlemen Association Beef Enhancement Funds. Currently, unfortunately, this is not being funded, which is a shame because well, as I will show you, that site has been heavily instrumented. And there's at least 4 or 5, there's actually a 5th publication that was just published related to this study. So everything that I'm showing you today has been published. And I'll be happy to share this references with you at the end of the presentation. But anyway, what we want, our main goal was to establish a long-term instrumented field trial, because most of the previous studies in Florida were based on laboratory and short-term studies under control situations. So like at the rainfall simulation, that study that I just mentioned, we can answer certain questions in a control environment. But if you want to really understand biosolids P dynamics under real world conditions, you have to do field trials. Of course, when you do field trials, there's a lot of things that you cannot control. So there's a trade-off when you do laboratory or greenhouse versus field study. But it's important to have that range of from a control situation to a more realistic situation. And that's the goal of this field trial. Of course, field trials are expensive, difficult to maintain, especially when you're talking about biosolids. Biosolids is not the most pleasant thing to work with, but that was our goal. It's the only field trial currently in place in Florida. And we want to evaluate not only the environmental impacts, but also the agronomic benefits associated with application of biosolids. So like I said, this is it was established in 2016. We're continuing the study. The first 4 years of data has been published. And I'm going to share a bunch of pictures here. So what we are doing in this field study, we have 3 biosolids types. One is a class AA, 1 and 2 are class B. We are applying them at the same nitrogen base rate. And in addition to the biosolids, we thought, well, what would be some strategy? And we knew that if we apply at a nitrogen base rate, we know that we're going to create excess P. So we decide to use another waste material. In this case, is biochar, and co-apply biochar with biosolids. And the rationale for that is maybe biochar can help retain some of the excess P, either improve plant P uptake or increase soil P holding capacity. So our hypothesis was that by applying biochar, co-apply with biosolids would have a less risk of phosphorus losses. Why we chose biochar? Because it's cheap, it's also a waste material, it contains no hazards constituents, and there's a number of very extensive scientific literature demonstrating that biochar can have benefits in terms of soil physical, chemical, and biological properties. Again, going back to our extremely sand soils, we thought there could also be some unintended benefits associated with biochar. Whether that was true or not, you'll see in the results. That was our rationale when we established this study. So just a quick layout of the plot. So we have 40 plots, 20 of those 40 are heavily instrumented, and I'll show you what the instruments are. So each of the plots, each of the 20 plots that had this red star here have a drain gauge lysimeter, and I'll show an actual picture of the lysimeter. We have soil moisture sensors because we want to see if is there any benefit associated with biochar or biosolids in terms of soil water holding capacity. We also have static chambers to measure greenhouse gas emissions. So when I mentioned in the beginning of the presentation, how biosolids can reduce or can provide a greenhouse gas, can be a greenhouse gas mitigation strategy. We're measuring that. And we also have pressure transducer. What I'm not indicating here, we also have suction cups at a different depths. So in fact, my schematic representation is missing a couple of instruments. So, but what I want to say is that each of those 20 plots are heavily instrumented, so we can monitor phosphorus movement, vertical movement in the profile, as well as some of the gas emissions when we think in terms of nitrogen. These are, I'm not going to get into details, the characteristics of the material that we've been applying since 2016. And some of the responses we are measuring, like I said, is not only the environmental aspect, but we also want to look at the forage production yield. Is there any concerns in terms of imbalances in nutrient concentrations in the forage that will eventually be grazed or cut as a hay? So now I'm going to show you a bunch of pictures, help you understand what the site looks like. Every year we do, we collect cores up to 1 meter just to monitor any nutrient movement in the soil profile. Here's a picture of the site when we're establishing, and this is the drain gauge lysimeter that we use for water quality monitoring. I believe this is actually, well, let's see, hopefully we'll play. When we instrumented. So those drain gauge lysimeters are installed at about 1 foot from the surface. It's just a quick demonstration. Anyway, and we have 20 of those. Soil moisture sensors, like I said, we're interested in looking at, is there a potential for these carbon-rich materials to increase water holding capacity? And like I said, so this is the grad student that was working on this project. It takes a lot of courage to work with something that can be quite nasty. These are plots that received the biochar. So biochar application proven to be something not very easy. So I cannot even imagine this in a commercial operation. It was very difficult. So we start seeing some issues, limitations associated with biochar right from the beginning. Just the land application was extremely difficult. This is us, the student. This is myself. This picture here in the middle. So nothing that I would miss doing. I'm not interested in repeating this experiment for another year. So again, some practical limitations associated with that. I see Dr. Sharma showing, that means that I'm running out of time. What I want to show you, I think that the bottom line here is the water quality data. That's the main concern. And I want to, this is nitrogen, but let me move to phosphorus, which is the main concern. So these blue arrows indicate when the materials, the different biochar, or I'm sorry, biosolids or inorganic fertilizer was applied. And the y-axis here is soluble reactive B in leachate. You see those spikes in soluble reactive B, so leachate B. It's usually associated with inorganic fertilizer, and usually happens either right after the sources were applied or during the times of the year where the water table is near the surface. So in South Florida, during the summer months, July, August, the water table approaches the surface and it stays at the surface for a couple weeks, many years. So that's when all that nitrogen pulse, I'm sorry, I said nitrogen, but it's actually phosphorus, nitrogen as well, but in this case, it's phosphorus. So basically the bottom line, the whole conclusion, and you can see this is data from 2016 to 2018. We continue to monitor the same data. Our conclusion was that inorganic fertilizer result in greater phosphorus losses, and there's no differences between biosolids and control treatments. Control meaning that treatments that received no P application. When you look at the percentage of phosphorus and nitrogen that was lost in this from these treatments, significant greater proportion of, let's look at nitrogen, for instance, about 47% of the nitrogen applied was lost. And what I didn't mention here before in the previous slide, I didn't show any biochar impact, because biochar did not affect, did not reduce nitrogen or phosphorus leaching. In fact, the biochar material that we used contain a lot of phosphorus, and it actually contribute to phosphorus leaching. So it was not, that particular material that we tested for 3 years was not a good sorbent. So it didn't provide any agronomic or environmental benefits. It didn't increase production, it didn't increase water holding capacity as many other studies claimed. In our case, we didn't see any benefit associated with biochar. So this is just a summary, the conclusions, and I just repeating what I just said. Unfortunately, we had high hopes for biochar to work in this scenario. We use a generic commercial biochar material that was available in Florida, and we didn't see any benefit associated with that material. But in terms of the biosolids, again, the results we obtained suggest that biosolids can be a viable source of nutrients with minimal impact on the water quality. So with that, I'm gonna end, and I'll be happy to take any questions. I'm sorry if I went over a little over the time.
No, you're perfectly all right. Thank you for your presentation. I really enjoyed it. All right,
Thank you.
We have 2 questions. Dr. Silveira, was the proportion of phosphorus in each sample evaluated prior to conducting the leachate and runoff study so that you know what each sample started with? Would this influence the amount of P found from leachate and runoff for each sample?
I'm not sure if I understood the question, but yeah, we know how much the phosphorus load, we know how much phosphorus we are adding with the different amendments. So we can calculate the proportion of the added P that was lost. And of course, also, I imagine that you are referring to the field study. Another important factor that will dictate the extent that phosphorus can potentially move out of the system is the soil P holding capacity. In this particular soil, it was not our intention. We would just want a typical spotosol with bahia grass, mimic a typical scenario where biosolids will be land applied. And it happens that this particular soil had a relatively high P holding capacity, which I think also explains so I went over that table very quickly, but less than 1% of the P applied via any source, including inorganic fertilizer, was actually lost via leaching. So that means phosphorus doesn't disappear. Either it's taken up by the plants or stays in the soil. In this case, the plants took about 20 to 30% of the applied P, but most of the phosphorus stay in the soil because of that relatively high soil P holding capacity. So if I were to repeat the same experiment in a soil that has less P holding capacity, my results will likely be different. Don't know that answered the question, but.
All right, so where was the field study? Was there a site where biosolids were already being applied historically?
No, the site where we established had never received any P fertilization, so never received any biosolids application, and during the past 20 years has not been received any inorganic fertilizer. So currently, well, there's sites in Florida, commercial operations that have long history of biosolids application, but we're not currently doing any field research on those sites.
All right. Did you compare different biochars?
We did, not in the field study. We did a greenhouse and a lab study, compare different biochar materials. None of them actually prove, well, the companies make a lot of claims. Well, they can do this, they can do that. Unfortunately, in terms of nutrient holding, especially nitrogen and P, we didn't. That doesn't mean that we didn't see any positive impact. That doesn't mean that all biochars. There's some materials that are engineered specifically for the purpose. So we evaluated in one of the greenhouse studies, we evaluated 7 different materials. None of them provided any benefits in terms of reducing nitrogen or phosphorus leaching.
Thank you. The follow-up question with the different biochar is, did you mix the biochar into the soil or it was surface applied?
In the field study, it was surface applied, but we also have a column study. So we have small PVC columns that we surface apply the biochar, and we act in treatments that we mixed with the soil column because we thought that maybe this lack of physical contact. Again, no benefit regardless of the method of application. Okay. I'm not trying to be pessimistic here. That doesn't mean that because I didn't see the results, the positive results, that doesn't mean that other materials cannot provide the benefit. It's just that those materials that we evaluated, unfortunately, didn't provide any benefit.
Thank you. We have a question. Did you incorporate the minimums? If so, how? Okay, so you didn't incorporate in the soil. So
no, in the field study, no, because it's a perennial crop that no, we wouldn't incorporate in a real world application.
Just a follow-up question on that. So Maria, do you think incorporating, like you kind of expressing that there may not be an impact of a method of application, but do you think this, if it would be soil mixed using a rototiller or a disc, would that basically let soil to fix more phosphorus because there will be more surface area that will be in touch with the biosolid?
I think so. I didn't mention, I had another study that we ran a few years ago, then it is also published. Instead of biochar, we use aluminum water treatment residuals, and we surface apply, and this was a field study, and we incorporated. Of course, for incorporating a pasture, you lose productivity during that year because the grass cover never recovers during that year. But there was a benefit associated with incorporating. So yeah, if you have, depending on what nutrient you were talking about, maybe for nitrogen, the mechanism controlling nitrogen, the interaction between nitrogen and biochar is different. But phosphorus, if it requires physical contact between the phosphorus and the biochar molecule, then incorporation could have. We saw that with aluminum water treatment residual. When we incorporated, we retain more P, and leachate P was less. So yeah, I suspect it could be the same. But the issue with biochar is that even when we run in the lab desorption studies, just equilibrating P with the biochar, they provided no sorption. They had no P sorption capacity. So in that case, it wouldn't matter if we're incorporating or if we're surface applying, because the material cannot retain any P. In fact, some biochar can be produced for any feedstock. Some that are produced from grasses, for instance, they can actually act as a source of P instead of retaining P. So it's very important. We call biochar this anything that goes through a pyrolysis, but the characteristic of the biochar, I think, will be very important to determine whether or not there's any benefits.
All right, we have 1 minute. And the last question is, did you perform any assessment of impact of heavy metals contained in the biosolids?
Heavy metals has been an issue that it's always been brought up. And it used to be a concern in the 70s when the federal regulations didn't have specific requirements for heavy metals. The current biosolids that are produced in the United States contain very little to none detectable levels of heavy metals. So I didn't mention we measure heavy metals just because there's that perception that could be an issue, especially if we're grazing, using animals to graze the plants. So we have the heavy metal concentration in the plant, but it's way below the ceiling limits because the material has a very low heavy metal concentration. But yeah, we did evaluate that, but just for sake of addressing that perception, public perception that heavy metal could be an issue, but it's not.
Thank you, Maria.
Can I just say, I'm sorry, very quickly, if you are concerned about the heavy metals, if you want to learn a little bit more, take a look at the report that I mentioned from the W4170 members. They talk about heavy metals in biosolids. So it has a very extensive literature on heavy metals and biosolids for current biosolids produced in the United States.
Thank you. Thank you, Maria. If you can hang till end of the session, that will be great. There may be a question. So I would invite Dr. Yiannis. I'll go by the first name, Yiannis. I'm sorry, I tried when Maria was trying to present, I was trying to pronounce your name. So I'll go with Yiannis. Dr. Yiannis is assistant professor, precision agriculture and smart machines in Ag Biosystem Engineering Department, IFS. Yiannis works most of the part in technology and artificial intelligence. So Yiannis, the floor is yours.
Thank you, Leachate. Yeah, today I'm going to focus a little bit more on emerging technologies and on artificial intelligence and how we can use AI and the new technologies for best management practices. So sometimes I like to start with this slide, and with this one, I'd like to show how AI, automation and robotics can transform agriculture to agriculture 4.0. So in the last few years, we had 3 key points that they totally changed agriculture. As you can see here, the first one was the introduction of machinery, mechanization, tractors, harvesters. The second was the introduction of chemicals like pesticides, herbicides, fertilizers, then genomics and genetics and hybrid crops. And now what's happening, we see how the new technologies like artificial intelligence and automation, robotics, Internet of Things, big data, how they can help us develop smart machines, and how these smart machines now, they are going to help us with precision application management. This is what I'm going to discuss today. Before I do that, you see here 2 examples of big companies like Google and Microsoft. Google, both of them, they develop and they invest on such technologies for agriculture applications. So Google with this, go back with this mineral program, they have this inspecting robots, and Microsoft with the farm beats. And also recently they got, I think, around $50 million with NSF to deal with the next G generation of communication. So solve communication issues, cellular communication issues, even space communication issues. And that will also help agriculture, where we collect a lot of data, but it's very difficult to transfer the data, especially real time, again, when we are talking about remote areas. So let me start with harvest technologies. I know harvest technologies not so much from best management practices, but I think here they provide a very good example of what we're going to see in the future. So again, from the left to the right, you see 2 technologies developed for apples or fruit crops, one from Abundant Robotics and the other one for FF Robotics. They use different technologies here. Or this is a linear actuator, like a relatively low-cost system and simple system. This is a vacuum-based that sucks somehow the fruit. And then we have a technology for strawberries from Agrobot. And of course, down here, you see a system developed by Harvest Crew Robotics, which is a local company actually located in Tampa, Naples, and a lot of UF professors, including my program, collaborating with this technology to improve this system. Again, this is more like a factor in the field than just a simple device. As you can see here, they grab the leaves. We play this video again. I don't know why that stops. They grab the leaves to relieve the berries. They use machine vision, AI to pick only the right berries. And as you can see also in this video, again, all these technologies, they have similar things in common. So like they use AI not only for detecting fruits and to pick only the right fruit, but also for navigation, for secondary inspection and quality control, even for packing in the field. And you see here these robots dancing. Some of these technologies, they also work at night. I think this video here from Harvest Crew Robotics demonstrates that. If I'm not wrong, I think I included that here, which is a huge benefit, especially, we need systems like that to address the labor shortage issue, which is very important. You see here again, this video, it's very impressive how this system can also work at night. So it can, in theory, can work 24/7. So as I said, we collaborate with Harvest Crew Robotics, and we have several small projects. In one of them, we use machine vision and AI to detect these raised beds. As you see with strawberries, we have double beds and find the center of the bed, and then detect the distance of the center of the bed to the robot, so you can properly align every robot above the plant to faster, to optimize this harvest operation. And also we have a PhD student that is developing a secondary inspection system with Dr. Lee from my department. And as you can see here in one of your projects, he tries to develop a machine vision and AI-based system for bruise detection. And you can see here we have some bruises, and this is what the AI detected. And something is happening here. We have one bruise here, one here, and one here, I think, and this is, you see different colors, you see all these detections. So this can be, when they harvest the fruit, they transfer them to the second level where they can have this secondary inspection system and also pack them in the field. But before you properly pack them, you need to detect the size, the color, and of course, any issues like with bruises or with diseases. So more about now best management practices and more about how we can use all these novel technologies and especially artificial intelligence to optimize some of management tactics and use them again for best management practices. So I will focus again for nutrient, pest, and disease management. So starting with weed management, here I have 3 systems. This video doesn't play, I don't know why. There are 3 technologies developed for precision weed management. As you can see, one from Blue River Technology. The second one is from Carbon Robotics. That for a reason we cannot see the video. And the second is developed by my program at the University of Florida. So all these technologies, again, they have something in common. They use AI and machine vision to detect the weeds and then treat individual weeds. So traditional sprayers, they spray everywhere. They spray in the crop, they spray on the weed, and they spray also locations that we don't have either of them. Carbon Robotics, they utilize a different technology. They use powerful lasers, and let me find the. So as you can see in this video, the lights that you see down here, they're actually lasers. So they still use machine vision, they still use artificial intelligence, but in this case, they use laser to kill the weeds. The other 2 technologies, they use chemicals. And what we found and what we see from other systems, using technologies like that, so you can improve, reduce the use of chemicals by 70%, sometimes even 90%. So this can be a good example of BMP, right? You can use a smarter technology, and again, I call it smart because it sprays only where we need it to spray to reduce the use of chemicals, and that means reduce also water, reduce a negative environmental impact, And also with weeds, like all other pests, we have a huge problem. We know that a lot of, we cannot kill all the weeds. They develop some kind of resistance, right? So I think we know more than 260 now weeds that they have some kind of resistance to the chemicals. And to develop new chemicals can be very time consuming and very, very expensive. So technologies like that might help. And this is the future. Some of them also, they are fully autonomous. As you see this one, also in my program, we received a grant from National Robotic Initiative, NSF grant to develop a fleet of robots to do this job. And this is the previous design. Now we have a new one. We are expecting to have the first robot ready by the end of this year. In a different project for now insect management, integrated pest management. In this case, we know that based on some global standards, and I think this is part also, or should be part of the best management practices, before we spray to control diseases or for insects, we need to have a region. So we need to monitor, for example, the insect population. So the integrated pest management, the idea is not to really kill the entire population of the insects, but we can control it and especially have this population under a specific threshold. So we need to develop a system that we can monitor the insect population in the field. And if it's above a threshold, sometimes the threshold is just only one detection. But in general, we have this threshold. If we detect more insects, like above this threshold again, then we can go and spray. And there are different methods to monitor insect population. That we have some stationary traps, like yellow traps or different other systems, but most of them are very time consuming, or they only catch insects that they fly. And we put them in different locations. They don't really collect a good sample. We don't really know what's really happening in the field. So one common technology for Asian citrus Sealit, which is the insect that transfers HLB, citrus greening, is this tap sample method. So here we use a stick. As you can see in this diagram, we take the branches, they have a paper sheet, and then to collect the fallen insects, and then we count only the adult insects. We put in a notebook, and then we go back to the office. We import the data to the computer, which is somehow quick, but still very labor intensive. And especially doing that in Florida, it can be very hard. So we try to automate that, and we have a paper. You can find more information about how that works. You see, we have this system, automated system is not exactly an arm. We can shake the branches. Again, this is a sample method. So we don't need to visit every tree. We shake some of the branches, and there's a platform with these blue boxes. They have cameras, low-cost cameras, to collect the insects and analyze them in real time using artificial intelligence. And also we have a patent about this technology. So again, this is an example of how a smart technology can help us for best management practices. Here you see the detection of some of the insects. And at the end, we develop a map. All these green and red colors that you see here, there are trees. And the red ones are the one that we visited. And you see with the numbers, the numbers of insects that we detected. Again, this distinguishes Sealits from any other insect and count only adult Sealits. Again, you can find out this video and more information about all these technologies in our Twitter. And of course, there's a paper, and also we have a patent in this system. So recently we submitted another proposal to National Robotic Initiative to automate that, to develop, not only automate, but develop robots to do this job. And actually we combine that with flash detection. We know that sometimes it's very important to connect the development of the flower and of the tree, of the plant in general, with the insect population development or growth. For example, with Sealits, we see that they are very active when we have new flowers, and this is exactly when we need to spray. So with this new system, we try to include this insect monitoring system, but also we have another technology that we propose to develop to monitor and detect new flaws. And also we have here a sprayer, but this sprayer is more like about emergency situations. This is how we propose it. And of course, here will be a cabin to protect the driver. But the idea is to develop, as you can see here, this multi-purpose robot to do this job. And it's multi-purpose because you can change the configuration. And I'm not going into details of the components that we have here. We have even a very novel camera to deal with issues with solar illumination and other stuff. And also we have a very novel navigation protocol that we're proposing. Very briefly to say that nowadays we see more autonomous vehicles in the fields, more robots in the fields. And for example, we see a lot of cars, right, from several companies that they have this capability. A lot of people believe now that these systems, they become very selfish. And to give you an example, for years now, we had this philosophical question that imagine that you are driving a car and from one side you have a hill, from the other side there are other cars coming, and maybe you drive with 50 or 70 miles per hour, and then two kids jump in front of you. And you have only two choices, right? One is maybe to try to brake, but you can potentially kill the kids, or turn right and go through the hill, or even maybe turn left and maybe have a bigger accident because more cars coming from the other side. So this is a philosophical question. We never had to really answer that more than to discuss about it. But now engineers, they have to really answer and take a decision because they can be a robot. So the robot, we need to program the robot to decide what to do. And what we see, or what a lot of scientists claim now, that we tend to take decisions that are more selfish. So try to protect the driver than, first of all, than anyone else. So in our case, we follow a different approach. If you can see here my laser pointer, this is a robot. It has to start from here. It has to go to this tree to collect data. And the easiest path is just to go straight away. And here we have two workers working the field at the same time. But if we go with this path, which is the fastest, we can like you see, we can go through the workers, and that might create other issues. So another path is more courteous path is to go around them. And then again, if you see in a tractor, maybe even turn here and come like follow this black line. And this is a novel approach that we're proposing, that we are going to consider workers and of course other machineries that they work on the farm at the same time. A little bit about drones, and I go a little bit quickly now. We start working with UAVs, unmanned aerial vehicles or drones, to collect data and try to see how we can use this data for best management practices. And some good examples here is we can detect diseases, but also we can use them for nutrient management. So I moved here in 2017. I started working on that in 2018. They took the first two papers, we published them in 2019. You see the first paper is about how we use AI to collect data from every plant. So the goal here is to develop a system that we can detect and identify every plant and collect data again from individual plants. We go down to the plant level, which we call phytotechnology. And then we use the system in this paper to evaluate several rootstock varieties in a large-scale experiment. So I'm not going in the details about how, what type of AI we use and how that works. Very briefly, again, to say that the goal is to detect the plant, start collecting data, and convert the data to practical information. And what this information is, for example, we can measure plant volume, plant height and canopy size, leaf density, which is also very important, health or stress level, and also we can determine plant nutrient concentration, among other data. And also in 2019, we developed this platform, cloud-based platform. We call it AgroView. And we received recently a lot of awards about this platform. And we have two programs, USDA grants that they support, partially support the development of this system. And also we have some grant from NVIDIA that help us to commercialize that. And we form a company. There's a company now that licenses this technology, agricultural intelligence, so it can make that available to the growers. And the goal here is like we identify a gap on these technologies, and actually it's more about on the data processing. Usually with drones, we collect a lot of images, thousands, it can be thousands of images from the drones, right? And then we need to use maybe 1 or 2 or 3 different software to create a map. We call it orthomosaic map. So we stitch these images together and then analyze these maps, because the map, it's a nice map, but what you can really do with a map, you need to convert again the data to information. So we developed this platform to automate all these steps, and this is how it looks like. So most of you probably are familiar with AgroView. We have also a paper in computers, electronics, and agriculture that describe how we develop this technology. And as you can see here, this is an example of a citrus field. It's around 2,000 acres. A grower used it for different purposes. I will explain soon. When you have it above every field, you see some Elysian analytics, total number of trees, total number of gaps. And then when you click view block or double click, you go inside. So inside the field, you can zoom in, zoom out. With all these dots, you see 3 detections. The blue dots are gap detections. Gaps are places that we had to remove the trees because they are not productive or dead. And again, here it's height is selected, so we categorize the trees based on their height in this histogram. And there's a video that a little bit demonstrates how the system works. Again, when you hover about every block, you see here even the pie, the ratio between trees and gaps. And as you see, we had a lot of gaps in this area. And then when you click view block, you see the map coming up. And then it's again very interactive. You can zoom in, zoom out. You can unselect here all these parameters. So here we can go back and select trees. You see all the trees. Again, height is selected. So in this histogram, you see height categories. And then when we click the gaps with all these blue dots, you see how many gaps we have here. So this grower uses technology to identify all the gaps and then order new trees for replanting. And again, we go about every of these categories. You see the total number of trees. We can change the bins, the categories. And here, for example, you can see also we can choose to display only trees that are then less than 10 feet tall. Again, you can find this video in our Twitter. And this is the plan, the fertility, development of fertility map. So this is the nutrient, for nutrient management. So what we do here, first of all, we determine plant nutrient concentration, again, for individual tree. But then we use different techniques to create these management zones because it's very difficult to present data per tree. So we decide to develop these management zones using interpolation techniques. Here, potassium is selected. Again, you see here all these, the zones from deficient to excess. And this is the range for all these zones. This is based on IFA's recommendation, again, based on best management practices and IFA's recommendations. So if we change, IFA decides to change these ranges, we are going to update this system too. So here in this field, you see a deficient zone, then a low, then an optimum, and this is a high. So if you see here the tree ratio, this is, I found that very interesting. So if the tree ratio means that if it's 100%, means that it's entire covered with trees. If it's less, it means that we have gaps. And now as you see here in this zone, the deficient zone, we have 60%, this is the tree ratio, and then the low 82, which is significantly lower than the other two zones. So that means that there's, in this area, there's some problem. Maybe it's with the soil, maybe with something else. And this is only for potassium. And when we choose phosphorus, we choose a similar pattern. So again, here we develop also the variable rate fertilizers. So my program already developed a variable rate sprayer and smart sprayer. I will talk about it in the next few slides. But also we develop a fertilizer applicator that it can read these maps and it can apply maybe more in these two zones and less in the other two. And this is automated because it has a GPS, knows that it's in this zone, and we can define how much we want to apply. And this is a very good example of technology that we can use for best management practices. And this is the a sprayer that we used in order to develop a smart sensing system and to optimize the application, convert it to variable rate. Very briefly, I would like to say that there are commercial systems that growers use right now. For example, the eye system to control zones. So as you can see here in this sprayer, we can split it in 4 zones from the left side and 4 zones from the right side, and every zone has, let's say, 3 nozzles. And they use sensor here, an object detection sensor like infrared, or it can be laser or any kind of object detection sensor to detect if something is here, turn this zone on. If you see something in zone 2, turn this zone on too. If there's nothing in from the right side, turn them all off. So that can save some chemicals. Why we need to spray when we have gaps, right? The limitation of this system is like it detects only objects. You see, I say object, I don't say trees, because it doesn't know if it's a tree or it's a human or if it's any other construction. Also, it cannot detect leaf density, so it cannot detect dead trees. So we received a grant from USDA again recently, started January 1st of this year, and we have already developed the first prototype. We use sensor fusion. In this case, we use a low-cost LiDAR. The LiDAR will detect the height of the object, so it has detects multiple points. And also we have some two color cameras, and see here the field of view, and we use several AI models, first of all, to detect if it's a tree, then use the LiDAR information to control the number of nozzles. We can even have individual nozzle control. If it's not a tree, have all of them off. So if it's a human, for example, don't spray. And also here, very briefly, let me say we call it sensor fusion because we use data from LiDAR, from cameras, from a GPS, and also from a flow meter. And now we have added also a flow control valve. And this is how the system looks like. This is an older version. And this is an example of a tree. This is a dead tree that we know if we detect a dead tree, we don't need to spray here. And at the same time we spray, we collect other data. And this is an example of what we can collect, like we can count fruit. In this case, it's immature fruit, green fruit. And this is a graphical user interface. I don't need to explain that right now, but here you see some information about the camera and how they detect the number of fruits, and then the speed, latitude, longitude, and we can also control all these zones. We can have it to auto, or we can turn them on and off manually. We have, again, more GPS and LiDAR for my camera information. If they're on and off, we can also control that. And also here, what I want to say, we can have this spray buffer. We can choose to turn them on before and after the tree, let's say 10 inches before and 10 inches after, so we can be sure that we cover the entire tree. And there's a company already licensed this technology, and we are getting very close to commercialize it, the chemical containers. And here you see a video of how that works. So let me pause it for a minute to see. This is the right camera. So what you see here, you see the longitude, the latitude, you see how many fruit were detected and average fruit size. And also you see information about the tree. So the camera is here, so now it looks somewhere here, right? So it looks that this is a healthy tree and this is the height of the tree. And we see how we spray. So here, this is where the camera looks, because the camera is somewhere here. And this is just weeds, there's no tree here. What you see here is the tree behind. And you see here we have a gap and the tree height is 0. In this case, the action is do not spray. And as you can see here, all these nozzles will turn off. In this case now here, as you can see in the pictures, we have a weak tree. Here we can even change the rate that we apply. We can apply less. Again, the first prototype is ready. We tested it in different commercial fields with a lot of growers. And again, you can find this video in our Twitter and our YouTube too. And at the same time, we can develop these spraying maps and spraying heat maps and the path maps. With this red line, you see the path of the tractor. And then with the colors, the blue, there are less, the orange, there are more that we applied. Again, we have a flow meter where we know exactly how much we apply. And we use the AgroView software to visualize that better. And here you see also the average velocity, what's the flow rate, and what's the accumulated amount that we spray, how many gallons. And similarly, you see information about every tree and every fruit, sorry. And what I'm going to say here is something very important that we are doing at the background that, again, it's very, very critical. Like with all these systems, we collect a lot of data. So we use powerful computers or embedded systems, microcontrollers. They have very powerful GPUs that they can analyze all the data and convert, again, in the field, convert data to information. So we don't need to store huge data. Like even the LiDAR can generate 3D points, so huge amount of data. Imagine now we add on that images that we can collect every few milliseconds. So what we do here, and all these blue dots, they are just examples of images. We don't save all the images, we save few as an example. And here we have a variety, we have no fruit, that's why you don't see any fruit. And here behind you see the heat map for fruit detection count. And this is an example of how it could look like. But again, we only store the critical information. And that will helps a lot with the data processing, because again, what we found out is very difficult for growers to use any of the system if it's not user-friendly. Even for scientists, we hate sometimes to have like 2 or 3 different software and to like spend hours to analyze the data. We automate this procedure with this AgroView technology. And I will skip these slides. Just very briefly, I want to say that we use also drones for disease detection. And our goal is to detect diseases at the early stage. Again, I'm going to skip them just to say that we work with tomato crops, squash, and watermelon to detect different diseases and distinguish also between this disease, not only between healthy and infected plant, but between, let's say, healthy bacteria spot and target spot. I know I'm almost on time, so I will skip these studies, but again, we have, it is well published, all these technologies. We have several papers. Feel free to check on them and email me if you have any questions. My program also generates EDIS documents for example, here we have instructions on how to fly, step-by-step instructions on what you do before the flight, during the flight, after the flight. And also recently we organized this Technology Ag Expo that was in May 10th and 11th. And we had also a lot of speakers from all over the world, and especially a lot of speakers from the industry that they present all these new technologies. To give you again an example, the sprayer that I presented, it's can be easily qualified to be like a cost share, part of the cost share program, because it will help to reduce even water and of course chemicals. And we plan to have this expo now every year. Hopefully next year will be in person. This year was a virtual event. And here you can find more information about this is my email, my phone number, and this is the Twitter account. Please feel free to follow us here and see more information, more updates about all these technologies. And in the near future, when I have my next presentation, I can always try to add new developments and how we improve all these technologies. And again, thank you for your time and attention.
All right. Thank you, Yiannis. That was a great overview and a pretty in-depth presentation. I haven't seen there is some compliment for you in the chat box. I haven't seen any question yet, but let me ask you a question that I received last time from Vivek. That is it possible to to uh, to detect I would say to get some information from the soil parameters, like what we get it from plant in terms of vegetative index like NDVI that correlates pretty well with nitrogen and other nutrients as we are doing more work. But is there any indicator in terms of light or any other parameter or a medium that you think that you are using to detect some soil parameters, including soil moisture or some other stuff?
Yes, before I answer your question, let me say that most of the people are familiar with vegetation indices like NDVI, but we don't really use them. Sometimes they correlate with some, let's say, with nitrogen concentration in the plants, but we develop our own models that they take the raw data from multispectral cameras. We develop, like we use machine learning and artificial intelligence to develop, let's say, more advanced models, and they are more accurate models to predict these values. And yes, there's way also to use remote sensing and data from multispectral cameras to determine or detect some soil parameters. I'm not doing any studies on that right now, but there are different studies available about even how you can use in a bigger scale. If you don't want to have a higher resolution, you can even use satellite images, but satellite images will give you very low resolution. So it can be, for example, some maybe in kilometers level, but you can use also drones to do something similar. In our case, we're working mostly with vegetables and tree crops, citrus. We focus more on the plant nutrient consideration. But yeah, something like that, it's possible. And if any of you guys are willing to do a new study, because what we are releasing here, we're doing much more and we're developing more models. I'm slowly releasing some of these features when we feel comfortable that they're pretty accurate. And still we try to improve them, but we do really large scale trials to evaluate these technologies before we present them here.
Thank you, Yiannis. I will convey this to Vivek if he's not here today. Maria, I have a question for you. You have leachate that you collected from your study. Was there, and you mentioned about the water table, rising probably pretty close to the surface. Have you looked over these leachates all over the year or just for the study period? That's my first question. The second question is, was there any compromise because of the groundwater was interfering with your phosphorus readings?
Yes, good question. So we collected leachate anytime that leachate was generated. So it was throughout the entire year, but we can only, we can collect 4 water samples throughout the entire year, but leachate is only when you have enough moisture. So yeah, and then there was an issue with when the water table was above the depth of our drain gauge lysimeter. Basically, what we're measuring is groundwater P level. So yeah, there's an issue when we use that. The drain gauge lysimeter was designed for soils, was not designed for soils with a high water table like we often experience in South Florida. So yeah, that's one of the limitations. So in order for us to interpret that data, we have to look at the water table depth compared. There's no way we can just look at the leachate data without looking at the water table depth, because that would definitely influence our responses.
Yeah, the reason I'm asking is that we have one study going on that has similar issues, and because of that learning, we developed our study that I'm involved with as a hybrid approach, taking some water samples, but at the same time take soil sample and use a DSAT to model that to calculate the nitrate lichen. So I'm not sure which crop you're working, but that might maybe, you know, I don't know. The DSET doesn't include all the crops. It works on very few crops.
You are right. Yes. So that later, that's why that figure didn't show. We decided to install some pore water samplers to address that. Yeah, you get a snapshot. You don't know how much you have to. Well, I guess you can calculate the volume of water that's moving vertically in the soil profile, but the pore water sampler will only give you the concentration, not the volume of water that's moving. So yeah, I think that's one of the limitations when you work under, and it is when most of the flows, not only leachate, but also gas flows occur. And it's difficult to measure during those time of the year when the soil is underwater.
Yeah, Dr. Dukes has done a 5-year study on that, and he found similar results, and that's why he advised us to do a hybrid kind of approach, doing a soil sampling at the same time, do a leak drain gauge. Yeah, all righty. I'm not seeing any question, so I would ask Emily. Emily, if you can type or copy paste the link for the survey. We have a very brief survey, less than a minute, less than 60 seconds, that you can use to evaluate our presenters, as well as to improve our program for the future. I would also, okay, here we go. Emily, you are ahead of me. So here is the code for CC credits. So if you want, please take that as a to scan this on your phone. We will also send a hard copy, or maybe Emily, if you can attach the hard copy in the chat box, that'll work as well. But if anybody has any additional questions, please reach out to me. I will be more than happy to convey that question to the concerned speaker and get back to you what they have answer for you. You can use this link to, let me open this link if it works. Yep, it works. So you can use this link. This is a very small 30-second survey for your speakers and for the organizers to improve their programming. Please help us here to improve on this programming. And there is some adoption options as well. So if somebody wants to adopt, well, Maria just mentioned that the Biosolid may not be an option for adoption anymore. So that would be a good question for her to reach. You know, if anybody has any question regarding the adoption, Maria would be a good resource for that person for those kind of questions. All right, with that, I will say thank you both the speakers for your amazing presentations, and audience, thank you very much for attending this session. Thank you, Dr. Dukes and Emily, for helping me organizing this summit. With that, we will meet next week on Tuesday at the same time, 1 o'clock. Bye, Michael. All right. Bye-bye, everybody.